Hello friends, this video on excretory products and their elimination part 12 is brought to you by examfear.com. No more fear from exam. So now the question is, how is the urine concentrated? As I said, the urine gets concentrated mainly in the Henle's loop. So here we will try to see what exactly is the mechanism that occurs in the Henle's loop. So what do we mean when I say concentrated? Now, removal of water from any fluid makes it more concentrated, from any solution rather. So here in case of urine also, the more amount of water gets removed from the filtrate, the more concentrated the urine becomes. So there is a mechanism called counter current mechanism which is responsible for the concentration of urine, for keep making it more concentrated. Now this counter current mechanism drives the way the Henle's loop handles this concentration of urine. So counter current mechanism is the mechanism which we will talk about in the next slide, and the part of the nephron which participate or where this counter current mechanism occurs is the Henle's loop and the vasa recta. So one common thing between Henle's loop and vasa recta is that both are U-shaped tubular structure, and that actually is one of the important reasons why counter current mechanism occurs in these two places. So now let us talk about the counter current mechanism. So what is the meaning of counter current? So why is it called counter current mechanism? That is because the flow, now let us suppose this is the Henle's loop. So this is the descending limb of the loop, and this one is the ascending limb. Right? So now the filtrate comes from the Bowman's cap, from the PCT, that is proximal convoluted tubule, into the descending loop. So the filtrate flows in this direction, and in the ascending loop it flows in the upward direction. Now since the direction of the flow of the filtrate is opposite in these two limbs, that is why it is called counter current mechanism, as the filtrate flows in opposite direction. Okay, so that is one thing. That is why the name is. Now, in one of our previous slides, I already discussed that this descending limb is permeable to water. That is, it allows water to pass across it. Whereas the ascending limb is permeable to ions or electrolytes. So these are something which we have already discussed in our previous slide. So with the help of all this information, we will now try to understand the process of counter current mechanism. Okay, now few more things just to remember. Now let us say, now because of this property, what happens is that in the descending loop, water tends to come out. So water tends to come out in the descending limb. Whereas what happens in the ascending limb? In the ascending limb, the ions tend to come out. Now let us say the sodium ions tend to come out. So the ions are pumped out in the ascending limb, and water gets transported out in the descending limb. Now this transport of water takes place passively. So this water transport is passive in nature, whereas this transport of ions is an active transport. That is, it occurs at the expense of energy. So we need to provide some energy for moving these ions from the ascending limb into the interstitial space. Now this space which you see here, now what is there inside the tube? Inside the tube we have the filtrate. We all know that. And what is there outside? Outside in this region, what is there? It is nothing but the interstitial space. So this one is, not only this one, this portion, all these portion is nothing but the interstitium or interstitial space, whatever you call it, interstitium, interstitial space, anything is fine, right? And also, please remember the location of the nephron. So where is it located? We can say that till this portion is the cortex, because the Bowman's capsule, the PCT, DCT, they all are located in the cortex, and this portion is the medulla. right? So these are some of the things which we already know. Now the question is, what makes these ions to move out continuously, and what again makes the water to flow out passively? What causes this passive transport of water continuously? So that is one thing that we want to understand here. The other thing is, why do we call this as counter current multiplication? I mean, what is multiplying here? That is what we want to know here. Okay, so you got to know the basics, and you also got to know what is our agenda here. Now, in order to understand this, we will use one term here, and that is osmolarity. What is osmolarity? Osmolarity is a term which is used to denote the concentration of a solution. So osmolarity is generally measured or expressed in a unit called osmos per milliliter. Now, greater the value of osmolarity, more concentrated a solution is. Right? Let us suppose if there is a solution with osmolarity 100, and if there is another solution with osmolarity 200, that means this solution is more concentrated and this solution is less concentrated. Right? Now, what do we mean by less concentrated? That means this solution has more water and less solute. And when we say it is more concentrated, that means it has less water and it has more solute. So these are some of the basic things which you should know, so that you do not find it difficult when I talk about the actual process in the next slide. So these are some of the basic things, because I will be using the term osmolarity very often in the next slide. So I hope you are clear with the basics and you are ready to understand counter current mechanism, because this is a little complicated mechanism, and I do not want you to get confused. So please understand these basics here in this slide. Once you are clear with the slide, please go ahead with the next one. So now we are going to talk about the actual process of counter current mechanism. Now, in order to understand how the counter current multiplication takes place, what multiplies, we will take an example. Let us say that initially the concentration or the osmolarity of the solution here in the descending limb, the ascending limb. So this is the descending limb. So this is the descending limb, this is the ascending limb, and this is the interstitial space. Now this space, they are all interstitial spaces. Now let us say that the concentration in the descending limb, interstitial space, and ascending limb, everywhere the concentration is the same. Let us assume that. Let us say that the concentration is, say, 200. And let us suppose here also the concentration is 200. And here also the concentration is 200. So now what would happen? As I mentioned before also, that from this limb, that is the ascending limb, ions are pumped out. And this pumping out of ions happens actively. That means purposely we move them out at the expense of some energy. So some energy is provided here such that the ions are pumped out. Now, what do you think will happen if ions are pumped out? What are ions? Ions are nothing but they are the solutes. So, you are basically shifting solute from ascending limb to the interstitial space. Now, in the previous slide, I told you that more the amount of solute, greater the concentration. So, you are actually increasing the amount of solute in the interstitial space, and you are decreasing the amount of solute in the ascending limb. So, as a result, what will happen? The osmolarity or the concentration of the interstitial space will increase. Now, let us suppose that the value of the osmolarity increases from 200 to 300. So it has increased due to the pumping of sodium ions. And what will happen here? The osmolarity will decrease from 200 to, let us say, it will decrease to 100. So let us suppose it has decreased to 100. Now if you compare the descending limb and the interstitial space. In the descending limb, the concentration is 200 and here the concentration is 300. So there is a concentration gradient which is developed between the descending limb and the interstitial space. As a result, what will happen? These two will try to come in equilibrium. That is, both of these, the descending limb and the interstitial space, they will try to attain the same osmolarity. Right? So how can they attain the same osmolarity? Okay, we will see that. So let me write here the steps. So in the step 1, what happened? Ions were pumped out. Ions pumped out from ascending limb to the interstitial space actively. That is at the expense of energy. So what was the result of this? The result of this was that the osmolarity of the ascending limb became 100 and the osmolarity of the interstitial space became 300. Right? Now, the next step, what happens after this? Now, a concentration gradient is developed. Concentration gradient is developed between the interstitial space and the descending limb, because in the interstitial space it is 300, in the descending limb it is 200, right? So what they will try to come in equilibrium. So how will they come in equilibrium? There are two ways. One is the concentration of the descending limb should increase, that is, it should become 200, it should change from 200 to 300. How is that possible? The concentration will increase either if solute is added into the descending limb or if water is lost. Now solute cannot be added because the descending limb is impermeable to electrolytes. It will not allow the electrolytes to come inside. It will not allow the solutes to come inside. So the only possible option is to lose water. So the descending limb will lose water. So this will result in passive transport of water from the descending limb to the interstitial space. So water will start moving out. Now when water start to move out, what is the result? The concentration or the osmolarity of region D will increase. That is, it will increase from 200 to 300. But now please remember that this pumping of ions is happening continuously, right? So now this 200 will get changed to 300. Now, with over a period of time, this 300 is also flowing down, right? And it is coming to this side as well, right? So this 300 will start coming this side. Now, what will happen? Now, when this 300 comes here, now here the interstitial space is again at 300. This is also at 300, but pumping of ions will take place. So 300 will reduce to 200. And what will happen to this 300? This will increase to 400 due to pumping of ions. Now when this increases to 400, again here new 200s are coming, many 200s are coming, right? So another 200 will come. So there will be a concentration gradient again. Again this will lose H2O, correct? So this will again increase and again this will come this side. Now by this process, what is observed? The observation is that gradually the osmolarity of the interstitial space is increasing from 200 to 300 to 400 and so on. So it will keep on increasing. So as we go down the medulla, the osmolarity of the solution keeps on increasing. Now, due to this property, that is the counter current, due to the flow of filtrate in opposite direction, the value of the osmolarity of the interstitial fluid is increasing as we go down. This process is known as counter current multiplication, and this is possible due to the U shape of the Henle's loop. And because of this reason, a gradient is continuously created between the interstitial fluid and the descending limb, and as a result, every time there is a passive transport of water. So this pumping of ions will take place by active transport, and this water transport will happen passively, and this will be an ever-going process. That's because every time this moves here, this concentration gets increased all the more, and that is why as you go down, the osmolarity increases. Osmolarity increases as we go down. deep into the medulla. So this is the concept of counter current mechanism, and it is due to this mechanism that the concentration of the fluid increases. So if you see, this fluid came here at 200, gradually the concentration increased in the descending limb, but again when it came to the ascending limb, the concentration began to decrease. This 300 became 200. Similarly, the earlier 200 became 100. So gradually the filtrate tends to become dilute by the end of this ascending limb. But again, when it comes to the collecting duct, again in the collecting duct there is a lot of water coming out. So that also happens because of the same process, because this is also interstitial fluid, right? So that in the interstitial fluid, since the concentration or the osmolarity is increasing, a gradient develops here, because this collecting duct also behaves in a similar way like the descending limb. So here also there is a concentration gradient, and as a result water tends to come out, and therefore the filtrate again gets more concentrated, and finally we get a concentrated urine out here. So please understand the concept of this Henle's loop very clearly. First understand the basic things which I have explained in the previous slide, and only then please switch to this slide. I hope I have been able to explain this to you properly, because this is very, very important, and this is very, very confusing at the same time. Okay, so now that we have completed our discussion on the urine formation process, let us quickly have a discussion on whatever we have discussed so far, because there are so many things that we talked about. Now, in the overall process, what happens is that substances like the useful substances like glucose, amino acids, salts, water, they are selectively reabsorbed as the urine flows along the tubular part of the nephron. When I say tubular part, I mean the PCT, the Henle's loop, the DCT, and the collecting duct. So throughout this, a lot of reabsorption take place, whether you talk about absorption of water or nutrients or salts or ions. So all those things take place. Now, the amount of water absorbed depends upon the amount of wastes in the body and the amount of excess water in the body. As I said, water is required by the body, but only the right amount. So when there is any excess of water, that amount gets excreted. Now, whatever amount needs to be excreted will get excreted, and the remaining water will get reabsorbed. Now, once it comes out of the collecting duct, where does it go? It gets into the calyces, and from the calyces it gets into the renal pelvis, and from renal pelvis it will get into the ureter, and from ureter it will go to the urinary bladder. So that is the storage place of urine. So it gets collected in the urinary bladder till the pressure increases. Now it gets stored in the urinary bladder for some time, and then gradually when the urinary bladder is full, so you get that feeling of urination, and then it is passed out through the urethra. So that is the entire process of formation of urine. So now we have discussed till here. So all these things are clear, that how urine is formed in the nephron, how it reaches the ureter, how it reaches the urinary bladder. In fact, till that point it is clear. Now the question is, how do we know when to pass that urine out through the urethra? So how do we feel that pressure? So we will talk about that in a new topic called micturition or urination, that how the urine finally passes out through the urethra. Now before that, let us have a quick review. Now as I said, urine formation takes place in the nephron, and the nephrons are located in the cortex and medulla region of the kidney. So the nephrons are located somewhere here. That is the cortex and the medulla region. Now in this, the blood reaches here through the afferent arteriole. The blood gets filtered, so the filtrate passes to the Bowman's capsule. From Bowman's capsule, it passes to PCT, where maximum reabsorption takes place. Then it comes to the Henle's loop, where osmoregulation is maintained. From there, it passes on to the DCT, and finally the collecting duct. Collecting duct puts it into the renal pelvis. From there it reaches the ureter, and from ureter it goes into the urinary bladder, and then from the urinary bladder it is excreted out through the urethral opening. So please and review it once or twice so that you do not get confused about whatever we have studied so far. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.